Okay, everybody, now it's time to start. It's my great pleasure to introduce Professor Don Cleveland from University UC San Diego. So let me introduce his career briefly. So he did his Ph.D. work and postdoc in UCSF at the Mark Kushner's laboratory. He did plenty of very important studies from the end of the 1970s. So clones, so tubulin, and also named tau, and so very important. So the gene product as being familiar. And so then he joined the faculty in the Johns Hopkins University, so medicine, biological chemistry, and so actually, so I did my postdoc in there in 1989 to 1991, just next to his door at Craig University. But we had always doing so the floor meeting and also barbecue party, and also lots of the science and the fun we shared Hopkins. Then he moved to the UC San Diego, and so he still gives his the study of the neuron cells, their biology, and also the working so neurodegenerative. Because when was in Hopkins, so don't lab generated transgenic mouse overexpressing neurofilament. So it's expected result is that so enlarge the caliber of the axon, but instead the mouse develops the ALS like phenotype. Okay, so he deepened his studies of the pathogenesis of the ALS and also its therapeutics. Now he's the chair of the neuroscience, chair of the cellular and molecular medicine and UC San Diego, and he has been elected U.S. National Academy of Science and Academy of the National Academy of Medicine, and so he has received plenty of the prize, such as breakthrough prize in the life sciences. This is very the famous prize with $3 million, and also Raji and Fami ALS endowed award for innovative healing in 2022, and Wilson Medal and this year, and the Rainer Water Prize, and also this year. Okay, so he's also a consultant of Ionis Pharmaceuticals, and also the investigation on the how to develop new therapeutic method for neurodegenerative disorders and its related disorders. Okay, so please start.
So, yeah. So first of all, thank you for the opportunity to be at Keio University. I've enjoyed multiple visits to Japan in the past. First time at Keio, and thank you for remembering that past. I remember the young Okano. Yes, it's from those days at Hopkins. All right, and you pointed out something I should point out, which is that yes, I have been and have since about 2003. I have been and am a consultant for Ionis Pharmaceuticals, which will be a central component of some of what I'm going to talk about today. And the story really began with the recognition from human genetics that of the major neurodegenerative diseases, that where we knew the genes that were causative or contributory to disease, those genes are widely expressed, some ubiquitously expressed, not expressed in individual classes of neurons whose dysfunction in those neurons might drive the specific diseases. And even more so, it was from genetics was the discovery from Andy Singleton and his colleagues that if you inherit 3 copies instead of normal 2 copies of a gene encoding alpha-synuclein, normal gene, wild-type alpha-synuclein, that you will develop early onset Parkinson's disease. And that was followed by the discovery that if you inherit 3 copies of wild type, not mutated, but wild type amyloid precursor protein, you will develop early onset Alzheimer's disease. So there is too much of a good thing. And so in those cases, a therapy that would be directly on disease mechanism would be just to lower the production of those normal proteins. And that even more so broadly, since alpha-synuclein misaccumulation is found widely in Parkinson's disease, and of course APP and its products, cleavage products, A beta is one of the signatures of Alzheimer's disease. So we thought, okay, why don't we just develop an approach to lower the expression of a target gene within the mammalian nervous system. And of course, for the young folks, all of you would say, well, let's use an siRNA. But no, we're going to use an older method, a single-stranded DNA, which and a single-stranded DNA, and I'll talk about the properties of that in just a moment. But and then but then the question is, how are you going to deliver it? And how are you going to deliver it broadly through the nervous system? And so the answer is, well, we're going to inject it into cerebral spinal fluid, use the natural flow of that fluid to deliver it broadly. And indeed, the approach was to use a single-stranded DNA, typically 18 or 20 mers, that if you can get it inside the target cell so that it can diffuse into the target nucleus, it can pair with the target pre-mRNA, RNA or pre-mRNA, making an RNA-DNA heteroduplex, the RNA of which will be a substrate for an endogenous, intranuclear, almost ubiquitously expressed enzyme, RNase H, that cleaves the RNA, but not the DNA in such a hybrid. So it's a catalytic RNA destruction mechanism, which is really simple, relying on the presence of an endogenous, largely intranuclear enzyme, RNase H. Now, over the years, the medicinal chemists have modified the DNA backbone and the 2' and 5' positions of the ribose sugar to give what are today's antisense oligonucleotides, or designer DNA drugs, I prefer to call them, gives them some remarkable properties that I'm going to show you in a moment. Okay, so the problem, though, is if you inject these into an organism in the periphery, zero, I mean totally zero, gets into the nervous system. But you can silence any gene you want, and as long as if it's expressed, any gene you want in the liver, it is so easy. But in the nervous system, none. So in the examples I'm going to show you today, we're going to inject it directly into the cerebral spinal fluid, and it's going to be injected intrathecally near the bottom of the spinal cord. Now, the thing about this was that, and I hear it all the time now, well, everybody says, well, just use an antisense oligonucleotide. Right. The problem about it was that everybody knew that this could not succeed. And these DNA-based drugs are 10 times, well, 15 times the size of a typical drug, and they're heavily negatively charged. And there were all the textbooks, pharmacology textbooks, said there's no mechanism through which these things can get into cells in an efficient way. And the breakthrough, and thank you for noting the breakthrough prize, the breakthrough was we tried it anyway. Everybody said it would not work. And indeed, I said that too, but I was hassled by a local neurologist, Richard Smith, and he said, you should try this, you should try this. And he was just adamant and irritating too. And he finally, he also had encountered the chief scientific officer of Ionis and had hassled him. And he and I both almost simultaneously thought, we'll just do the experiment, show him that it fails, and move on. Okay. But subsequently, we now know that we can deliver these things very broadly, not just in rodents, but in non-human primates. And indeed, some regions, when you dose intrathecally, you can dose beautifully to the spinal cord, the cortex, you can get part of the hippocampus. There are regions that can be very efficient, and some regions that are much less reachable in these non-human primates, and almost certainly not reachable in humans. But the place where he started was in ALS, in inherited ALS from, and again, this audience knows selective death of upper and lower motor neurons. And a simple calculation is that there are a few, even though it's not a really prominent disease, there are a few million people now alive on the planet who will die from this disease. And it was the discovery of the first mutations that were causative of disease, dominant mutations in the gene encoding cytoplasmic superoxide dismutase, that led by consortium investigators worldwide, led by Bob Brown, who reported these mutations. And the News and Views article in Nature accompanying it declared, because we all knew that oxidative damage drives neurodegenerative disease, the News and Views article declared that these mutants were dominant loss of function mutants. And I looked at those mutants, there were 11, and none of them, and even now with more than 100, almost none of them are premature translation terminators that preclude the synthesis of a nearly full-length polypeptide, exactly the kinds of mutations that are easy to generate and that would be there if simple loss of function could drive disease. And indeed, what we did was we made, of those 11 mutations, we made 6. I chose them. I chose the 5 that had charge shifts so I could recognize the mutant protein, and the 6th that was thought to be the most common cause of human disease. And we showed that one of them was fully active as a dismutase, and one of them was fully inactive by every assay as a dismutase, and the others were in the middle. And we took one of those, Lucy Brune, when she was a postdoc with me, she took one of those, glycine to arginine at position 85. This is completely inactive in everybody's assay, has no dismutase activity. And she asked, in mice that have a transgene with an inactive mutant that causes fatal paralytic motor neuron disease, what happens if you lower dismutase activity by deleting one of the 2 endogenous alleles or both? And the answer is, what does that do to the disease course? Nothing. Nothing to timing of disease onset, disease progression. Dismutase activity is irrelevant to the pathogenesis, at least of this mutation in mice. And that led to the now, I think, consensus view that these SOD1 mutations provoke disease primarily, if not exclusively, through gain of a toxic property, not through loss of activity. Although in 2023, we as a community, I think, do not agree on what the toxic property is. Okay, so, but we could be sure that if you could just turn it down, this would probably be on therapeutic, a real therapeutic strategy. And this is the founding experiment done in a transgenic rat that has a hyper-aggressive disease course. So it's a transgenic rat expressing an ALS-causing mutation in SOD1. And sure enough, we could lower the level here in the yellow near the site. In this case, we're injected intraventricularly in the mice. And near the site of infusion, we can lower in a dose and time-dependent manner, lower the amount of SOD1 RNA. But we learned right in this founding experiment, wow, CSF does circulate, and you dose in the ventricle, but you see SOD1 reduction throughout the entirety of the rodent nervous system. And even more so, in this transgenic rat has, as I indicated, a hyper-aggressive disease course where untreated or saline-treated animals only live about 25 days after phenotypic onset. But in this founding experiment, dosing them with an ASO to lower production of SOD1 at disease onset, doubled survival after onset. So we thought, actually, well, we've really done something. And we then extended it to show that you could dose non-human primates, dose and suppress endogenous genes in those non-human primates. We thought, okay, we've established that this is now feasible. Went from, it can't possibly work, this is really feasible. And we sent it for publication. And this is what came back. So we sent it to Nature Medicine, and it came back editorially rejected. And no conceptual advance, because turning off a toxic gene would be clearly the therapeutic strategy, so there's just no conceptual advance. And then I got that the then editor-in-chief on the, I exchanged emails with him, and he said, well, he admitted that the paper is a nice demonstration of the effectiveness of the modified oligonucleotides, and I confess the experiment in the monkey is remarkable. Nevertheless, this is not a substantial therapeutic advance, as you'd have to inject the ASO. And I got him on the phone and I said, gosh, I thought we injected things in medicine. But, and he said, this will never be the basis for human therapy. And he then published 2 lentiviral integrating vectors encoding an shRNA, calling those therapeutic, each of which, including one from a company, was dead the day it was published by Nature Medicine. I subsequently suggested to this editor that maybe he needed a more descriptive name for his journal. And I said, so what could it be? Oh, and then I said, oh, I got one. How about Nature Non-Medicine? Anyway, okay, so and here's the founding, this is the founding paper where we demonstrated that both in rodents and in non-human primates, you could deliver these drugs effectively throughout the nervous system. And now, of course, we expected then to quickly get to clinical trial. Now, Ionis was uninterested in working in the nervous system. The preclinical toxicology of the first drug to go into man, they did not pay for that. I paid for it. We raised the money philanthropically. I and my then postdoc Tim Miller, we raised $1 million US dollars to go to enable the first clinical trial. And indeed, we showed that in the phase, there was an initial phase 1 trial. The drug was safe. And then Ionis learned how to make these even more effective with different modifications. And we went back to a second phase 1 trial, which in 2020 was reported. It was safe, and indeed, it appeared in the phase 1, phase 2 trial. If you compare the rapidly progressive disease patients in the dashed blue as compared to those placebo patients versus the ones that were dosed, we slowed disease course in the rapidly advancing ones, and the same is true in slowing disease course in the longer patients. And indeed, just in 2 years later, in the outcome of the phase 3 trial, what we see is that there was a slowing of disease course, almost a flattening of disease course in the second 6 months of therapy in the treated patients. And just last month, almost a month ago today, we've finally received the first FDA approval for use of an ASO in an adult neurodegenerative disease, where in fact, in talking to the clinicians, especially Tim Miller, that in 25% of the patients in this trial, dosed for 1 year, did not just slow disease advance, 25% improved in multiple clinical measures. So we have partial disease reversal in those indications. FDA did not look at that evidence because it came from the open label extension, but in fact, it's really quite, at least in all the, as a non-clinician, I talk to neurologists everywhere, and no one, especially in these rapidly advancing cases of SOD1 mutants, no one has seen disease reversal. Okay, so, and in the subsequent time, so now we know that we can deliver these things broadly, both in rodents and non-human primates. We can use them for targeting RNA degradation if the oligo supports RNase-H-dependent cleavage of the RNA, or you can modify the oligo so that the hybrid RNA-DNA hybrid is not recognized by RNase-H, and use them for altering splicing. And I didn't include the data today, but it's absolutely true that the best of these ASOs are really remarkably long lasting, so that you can get 3 to 4 months of efficacy from single injections. It's certainly commercially feasible, and in all but one of the trials that Ionis has taken forward, it's clearly safe. All right, so the founding success was in the childhood disease, spinal muscular atrophy. My team did no part of that, except for enabling Ionis to be interested in working in the nervous system by demonstrating that we could deliver these things safely and broadly in the adult nervous system. The there have been multiple trials. I talked about the trial in SOD1. There have been trials for the most frequent genetic cause of ALS, this repeat expansion in the C9orf72 gene. That trial failed. And a second trial from the company Wave failed last week. So there have been two failures where I think there's an intra-team disagreement with Ionis about why it failed, but this is one of our biggest disappointments. There are other ongoing trials in ALS. There was a Huntington trial. There are two Huntington phase 3 trials, one that failed in 2019, also two that failed from the company Wave. But Roche is conducting a second trial with excluding patients except for those that are very early in disease onset. It's a 350 patient trial. I think there still is a chance for success in Huntington. Now, and in Alzheimer's, there's a trial lowering tau, and in Parkinson's, two trials ongoing. One to lower the LRRK2 kinase, especially for those mutants in which there's been activating mutations, and in lowering alpha-synuclein, back to the original discovery that triploidy for alpha-synuclein is sufficient to cause early onset disease. We expect the phase 1, phase 2 readouts to be available in 2024. All right, but I'd like to.
So in case of vaccine 2,
yeah.
Do they select the patient with vaccine Ph.D. syndrome?
Okay, so actually, I would invite you to interrupt. Not that I could stop him from interrupting anyway, but you at any point. But so for ataxin 2, yeah, no, the patients used for ataxin 2 are sporadic patients on the strength that genetic evidence that Aaron Gettler and his colleagues had identified of an intermediate repeat length in ataxin being a potential component of disease. My understanding, actually, let me maybe correct, but amplify my answer. So I think they're taking sporadic patients. I do not think they're screening them for the repeat, an intermediate length repeat. But it's a good question, Video. I'll inquire further. I'm not 100% sure. It's definitely ongoing. Actually, and as many of you will know, when you're dealing with a big pharma that's now taking things forward, information flow stops. So I know Gettler is extremely irritated that he knows nothing whatsoever about what's happening in that trial. And indeed, that the Ionis model has been not to conduct the trials themselves, but to partner with big pharma to do that in every one of their trials, except now actually the fourth one, mutation and plus, that's now being conducted by Ionis. Okay, so but that reminds me, and earlier today there was a discussion, so I put these in. So yes, this is the 46th anniversary since the discovery of tau, and this young graduate student first purified it, and then described its chemical properties. Actually, we showed that it was a natively unfolded protein. We just weren't smart enough to coin the phrase. But and then of course subsequently it was discovered that tau misaccumulates into tangles inside the affected neurons in Alzheimer's disease and in chronic brain injury, and that the tangles of tau, at least for Alzheimer's, are today's best correlate of cognitive decline, better correlate than accumulated plaques. And indeed, this came up in this afternoon's discussion. Here's the purified tau from 1977. Wasn't so pure on an SDS gel. These are actually Kumasi stain gels. And so if you cut those bands off, you can see that there was 1, 2, 3, 4, 5, 6. All 6 tau isoforms were apparent in the original isolate. And there was a long discussion about whether I would ever graduate, since I was unable to purify the tau. And so we invented a method to use peptide mapping where you could cut bands off a gel and then layer a new protease on top of the second, put that gel slice on top of the second gel, and layer protease on top of it, and get remarkably stable partial proteolytic products as you have an increasing level of protease. And you could see then the tau isoforms, the various isoforms, gave rise to highly similar peptide maps, not identical, but highly similar with each of the two proteases. And so we concluded that it was a heterogeneous series of tau polypeptides. There were 6, as you can see there, 6 isoforms. We did not propose that they were the 6 alternatively spliced isoforms from a single gene. And why did we not propose it? The young folks are never going to get it, because splicing had not been discovered. Splicing was discovered in 1978, the year after. Okay, so, and just last month, the phase one trial to target tau in early onset Alzheimer's, in the earliest phase of Alzheimer's in mild cognitive impairment, that was published in Nature Medicine. I guess they are working in medicine now. Okay, it's a different editor. Okay, so a feature of that, perhaps not surprising, but here just to show what you can do, if you measure total tau that's released into CSF, probably from neuronal degeneration, you can see in a dose and time-dependent manner, you can lower the level of total tau or phospho-tau. Many think phospho-tau is the bad tau. Okay, so, but that's not the surprise. Here's the surprising part. It's kind of an amazing part. And Biogen released these data at the AD/PD meeting this year. Nobody noticed. And there are 2 patients using, there is a PET ligand to measure accumulated tau. Not exactly sure what kind of accumulated tau it is, but there it is. And here in this initial scan of this patient before the trial, you can see there were deposits on both sides of the brain and in the central region. And then this is a placebo-treated patient for the first 6 months. And you can see that the deposits, for example, the deposits on the left-hand side got redder. This patient was continuing to deposit this aggregated tau, imageable tau. And so was the second patient, where you can see on here on the right-hand side, those deposits got larger during the phase of placebo therapy. And then, after 1 year of anti-tau ASO therapy, what happened? And the answer is the deposits that were already there largely disappeared. Not completely, but largely disappeared. So the N is a little small. 2. This is the only data that Biogen has released, but it does demonstrate, I think, that at least for some patients, dosing over a time course of 1 year, you can lose the tau deposits that were imageable tau deposits that were there before. And I think it then portends well for future. Yes. No, so Biogen released no information whatsoever about cognition, a key component. You're absolutely right, and so it's I'm as I would be, as I know you would expect me to be. I'm cautiously optimistic, but of course we need a much bigger N, and we need the cognitive data completely. Okay, and of course for chronic brain injury, the definition of the disease is this misaccumulation of tau. You see tau deposits here, or in a higher magnification. These are from Anne McKee and her evidence in the United States. This is called National Football League disease because of its prominence in American football players. Anyway, so the phase 1 trial in AD is effectively the phase 1 trial for suppressing tau in National Football League disease. So I do suspect, especially if we see it when the readout comes in 2 years or so from the cognition in the AD trial. We may be in chronic brain injury. Okay, but this is supposed to be about ALS. So back to ALS. So the original discovery from 2 teams in 2006 that an RNA binding protein TDP43, which is typically like this neuron here, typically intranuclear, largely intranuclear, that in most instances of ALS and about half of frontal temporal dementia, it misaccumulates in the cytoplasm in this kind of aggregated form. And kind of obvious as well is that where it has accumulated in the cytoplasm, it's been depleted from the nucleus. So it looks like a combination of a loss of nuclear function and gain and potential gain of toxicity from the cytoplasmic deposits. We now recognize that that occurs, and I think there's a consensus view worldwide that occurs in almost all ALS patients, with the marked exception of the SOD1 mutant patients and the FUS mutant patients, but on the rest, and it's about half of frontal temporal dementia. All right, so we initially used an ASO to reduce TDP43 in an adult nervous system of mice, and we saw that 1,500 RNAs changed in abundance or in splicing pattern. So it functions as an RNA binding protein broadly, but we missed what we think is now the most interesting one. And we went and redid that, now using an siRNA to suppress TDP43, and not suppress it completely, but to suppress it here fourfold, and ask what happens to the other RNAs when you do that. And there's one encoding the protein Staffmen 2, which is suppressed even more than the level to which you suppress TDP43. We missed it in mice because the Staffmen 2 gene in mice is not regulated by TDP43. This is human selective. And now Staffmen 2 is the RNA, the RNA encoding it is the RNA most affected by reduction in human neurons, by reduction of TDP43. It's actually a relatively abundant RNA. It's the 20th most abundant RNA in human motor neurons. It's been proposed to be a direct binding partner of the alpha beta dimer, that is the subunit, that is the assembly unit for microtubules. Okay, so and here's the pre-mRNA from Staffmen 2. It's got, or the gene in pre-mRNA, it's got introns, exons, and what happens is that there is, if you lower TDP43 to a third, a quarter of its original level, the protein level of Staffmen just disappears. So again, at both RNA and protein levels. And if you now ask, so what happened to the Staffmen 2 RNA? And the answer is, well, it gets polyadenylated at a cryptic polyadenylation site that sits within what should have been intron 1 of the pre-mRNA. And indeed, there's an additional cryptic splice site. So cryptic splice and polyadenylation sites make a truncated RNA. And indeed, if you can see that truncated RNA by a classic RNA-seq experiment in cell culture. Here, if you have normal cells, you see exon 5, exon 4, exon 3, exon 2 with a 3', a 3' bias as you might expect for a typical RNA-seq experiment. But if you now suppress TDP43, you now have almost exclusive use of the cryptic splice and polyadenylation site. And if you look at the actual sequence, within that intronic sequence where the splice and polyadenylation sites occur, there are 3 GU-rich segments closely spaced that are the prototypic binding site for TDP43. They can't bind 3 TDP43s. You can only bind 1, but it's a predicted and validated TDP43 binding site. In human, but not mouse, Staffmen pre-mRNA. And indeed, it is really used in sporadic ALS patients, as we determined by taking laser captured motor neurons from surviving motor neurons from sporadic ALS patients, and now asking in that RNA-seq experiment, again, from normal individuals, you see exon 5, 4, 3, 2, just what you saw from the cell culture example. But in the ALS patients, sporadic ALS patients, it's almost completely cryptically spliced and polyadenylated to produce that truncated RNA that encodes 17 amino acids, a non-functional Staffmen protein. So this is a Staffmen loss of function induced by TDP43 loss of function. Okay, so so what does Staffmen do, and how does it do it? So part of that is still to come. But a lot of that is still to come. But it is indeed an axonal protein. It's an axonal protein seen in green. It accumulates at growth cones. It actually accumulates at neuromuscular junctions. So it could function in any of those roles. And so one of the questions that we just posed is, so if you had a normal adult nervous system, and you depleted it of Staffmen, what would happen? And we've depleted it in either of two ways. Transiently depleted using an antisense oligonucleotide to target selectively the Staffmen synthesis, or to use an shRNA that was delivered focally to lumbar level motor neurons, so that we can but starting with a fully normal aging adult animal, and now just depleting Staffmen, and ask what happens. And the answer is, if you do it for a long term, you get denervation. So Staffmen's essential for maintenance of mature neuromuscular junctions. Yeah, and you can see it actually gets quite striking denervation that in an age-dependent manner, but slowly. You start suppressing Staffmen synthesis, and it evolves over months, not minutes, not days, not hours, not days, but months. And if you suppress it transiently, you get that effect, but of course, to a lesser degree. So we argue that continued innervation requires Staffmen, and so that its loss in ALS is likely to be a component of the actual pathogenic process, not irrelevant to it. And so then we said, so how does TDP43 work to suppress the use of those cryptic splice and polyadenylation sites? And so what Michael Bond, my graduate student, did was to take this GU-rich domain that is the TDP43 binding site, the clustered sites, and he made a 13 or 14 nucleotide change to convert it from this GU-rich sequences to the binding site of a well-known bacteriophage RNA binding protein, the MS2 from the MS2 bacteriophage. So didn't change the spacing, just changed that sequence. And now he's going to ask, does TDP43 do something really special, or does it just sit there and bind to those sequences and then block the usage? And so the way he started that was he's built the we started by modifying an endogenous allele, a Staffmen allele, in these human neuron-like cells, and modified one allele with this MS2 loop, replacing the TDP43 binding site. And so now we could tether TDP43 not by its own direct binding to that site, but tether it indirectly by tethering it through the MCP protein encoded by the MS2 bacteriophage. Okay, so of course we're going to express that protein, force expression of it, and here's that design. And sure enough, if you park RNA binding incompetent TDP43 and glue it onto those sites by using this MS2 targeting sequence from an MCP, what you end up with is, oh, look, the level of this truncated RNA plummets by binding this component there. And in our heterozygous cells where the affected allele gives rise to no functional Staffmen RNA, yep, you can restore it. So we thought, well, we've demonstrated that TDP43 in an RNA binding independent manner does something there. But I admit, I had made the student do the control. And I'd made him just glue the MS2 coat protein onto the RNA, and that works just as well. So you don't need TDP43, but we know what TDP43 does at that site. It sterically occludes the access of either the splice or the cryptic polyadenylation site factors. And in work I didn't show you here, but we established it's the cryptic splice that matters. The cryptic polyadenylation site is not used if you mutate the cryptic splice site. So TDP43 sterically blocks the use of these cryptic sites. Okay, so now, can we use that information therapeutically? And I mentioned the success in spinal muscular atrophy before, but to just remind, the SMN1 gene is mutated in spinal muscular atrophy, and such that all patients are homozygously inactivated for the SMN1 gene. And actually, I didn't look up the Japanese population, but in the American population, about 1 in 35 individuals carries an inactivating mutation in the SMN1 gene. So that in random marriages, that's about 1 in 35 squared kids will be born with a homozygous inactivation of SMN1. But almost alone among the primates, we have a second gene, the SMN2 gene, that could encode the exact same protein as the SMN1 gene, but it's chronically misspliced to miss out exon 7. And the strategy that Adrian Craner had shown originally, and that Frank Bennett at Ionis demonstrated a therapeutically viable way to do, was to use an ASO to correct the splicing. And indeed, here, if you looked at cells from one of these individuals with the homozygous mutation or double mutations in the SMN1 gene, they indeed, from the SMN2 gene, they make mostly this RNAs that missing out exon 7, but with an ASO, you can convert that to mostly correctly spliced. And that led to, that was the first use of an ASO in the nervous system, for this childhood disease, where Frank Bennett and Adrian Craner also won the Breakthrough Prize in Life Sciences for demonstrating the efficacy of restoring SMA synthesis by using an ASO, these RNase H inactive ASOs that were spliced correctly. Okay, so now back to ALS. So if we know that when TDP43 levels are suppressed, there's cryptic spliced and polyadenylation of the Stafflin pre-mRNA, making a non-functional truncated product. We know it's seen broadly in sporadic disease. 13 out of 100% of the sporadic cases that we looked at in detail, 13 out of 13, we saw that. And so now we're going to ask, can we identify ASOs that can restore this, can block the usage of these cryptic splice sites and thereby restore Staffmen synthesis, even though we're not restoring TDP43 function. And we start with cells that we constructed, neuron-like human cells, in which we have homozygously modified the endogenous TDP43 alleles. I can say we did that intentionally, but we got both alleles modified, and so we homozygously did that. But on the strength of that, actually, there's partial loss of function from those TDP43 mutations, and you can see it by the relative accumulation of full-length Staffmen mRNA. So it's there's about half, and now we can use those cells to screen for ASOs that can suppress the usage of the cryptic splice site and producing the truncated RNA. And here you can see the blue ASO in a dose-dependent manner can suppress that. So can the red ASO. We tiled across the entire region here with ASOs, looking for the most effective ones. And then, even more important, can you restore the synthesis of the Staffmen encoding RNA? And the answer is both the blue one and the red ASO can do that. Actually, many ASOs can suppress the use of the cryptic site without restoring the full length one. Okay, so now, what if we now take cells, in this case, iPS-derived motor neurons in culture, and we're going to suppress them for TDP-43 and ask, well, what happens? What's the phenotype of that? We're growing them in compartmented chambers where the cell bodies are in one compartment. The axons grow across a ruled array. And then, as you can see, from the end of the ruled array, you've got the axons growing out here with their growth cones. And they certainly incorporate neurofilaments marked with NFH here, and Staffmen in green in the normal case. And now we're going to treat them we're going to axotomize them. Sorry, we're going to axotomize them and ask, can they regrow? And that's what I showed you in the top one. Yeah, you could cut them off, just mechanically shear them off, those processes, and they regrow within a day. Okay, and that's what you see at the top. Now, if you suppress TDP-43 with an ASO, they cannot regrow at all after this axonal injury. So now TDP-43 affects many RNAs, but what if we now just restore Staffmen using an ASO to restore the correct splicing of the Staffmen RNA? And the answer is, despite the fact that all of the remaining RNAs are still affected from the loss of TDP-43, regeneration after injury is almost fully restored by just restoring Staffmen back. So we argue from this that this is a likely therapeutic target. Its loss alone drives denervation, and that we can restore it here. We've just built humanized mice, humanized at the Staffmen alleles, to be able to, and we've demonstrated that the ASOs I've just showed you here can restore, in a humanized Staffmen gene, can restore the synthesis of a full-length Staffmen protein and the correct splicing, if starting from that MS2 modified Staffmen allele. So we believe that we have found lead molecules for which therapy can be done. I should have included today, but I just realized I left out. If you suppress Staffmen chronically in normal, the adult mouse nervous system, what happens? Well, there's denervation, but if you look at the axons, those proximal axons, motor axons, the axons shrink in diameter. They shrink and shrink, but they stay attached to the myelinating cell, thereby ripping the layers of myelin. And you have what looks like free-floating axons, shrunken axon, sitting in this broken layer of myelin. So we argue from that indeed that's and there is a reorganization of neurofilaments. They are not phosphorylated, and that is exactly what we see in human motor roots from ALS patients. So we argue that Staffmen is a good target for restoring innervation at the neuromuscular junctions and for recovering after other injury. And so here, the founding of Staffmen as a potential component of sporadic ALS appeared in 2019 in back-to-back papers, one from my group and my long-term partner, Clotilde Bagier-Turian, and Kevin Egan, then at Harvard. And indeed, both we argue that we have now, earlier this year, demonstrated ASO-mediated restoration of Staffmen in the mammalian nervous system. And we're expecting Curalis, a new startup company, went to trial in late 2023 with an ASO, and we expect Ionis to get to trial in 2024. Okay, so, and it just left for me to thank those who acknowledge those who really have been a part of this. This is the founding team for the use of ASOs in the human nervous system, starting with Tim Miller, who was then a postdoc with me and who has run the ALS trials for SOD1. He's also done the work demonstrating that you can suppress tau in mouse models or alter splicing of tau using ASO. Richard Smith was the neurologist in San Diego who insisted that we do this. And this is my mouse colony surgeon and colony coordinator, Melissa Macalonis. Frank Bennett, the CSO of Ionis. And again, yes, we just last month, the first approval in ALS for, and then for Staffmen, targeting Staffmen, here's this, the San Diego team, Yoni Lopez Araskin, Zeev Melamed, just now independent at Hebrew University in Jerusalem, Michael Bond, a pair of my graduate students, Michael Bond and Melinda Bakari, my long-term partner, Clotilde Agier Korean, now at MGH, the Ionis team, and Kat Lutz and her colleagues at JAX, who the Jackson Laboratory, who were instrumental in many of the mouse things that were done. And lastly, my former colleague now at the Leuven, who was responsible for the trial, ASO trial for FUS. She forced Ionis to find those ASOs. And without that, there would be no trial. Okay, thank you very much for your attention.